Good afternoon. I'm Stephen Peck, and today we're going to be covering the topic of oxidative phosphorylation. I'm sure we've known ever since preschool that if we don't have oxygen to breathe, we die. But that begs the question, why do we actually need oxygen? In this presentation, we'll answer that question. So, oxygen is needed for oxidative phosphorylation, which is a fundamental cellular process of creating energy from food. From hereafter, we'll call oxidative phosphorylation OP. So nearly all living organisms, almost all the energy coin molecule, which we call ATP or adenosine triphosphate, is created through oxidative phosphorylation. There are a few organisms, usually one-celled organisms like bacteria and yeast, that can create all the ATP they need simply through glycolysis. And we perform glycolysis as well, but in our case, it's not going to produce enough ATP to keep us alive. So before we talk about OP, let's talk about the citric acid cycle because it's closely linked with OP. And there's some components created in the citric acid cycle that we're going to need in OP. So here's the cycle. Here's a separate reaction, pyruvate oxidation that happens at the beginning, but here's the main part of the citric acid cycle. And we see these red rectangles indicating a molecule called NADH. Right here and here and here, and these are electron carrier molecules that are created and are needed for OP. Here's another electron carrier called FADH2 that's also used in the OP process, and we get just a little bit of ATP that's created during the citric acid cycle as well. And all this happens in the matrix, or the interior of the mitochondrion, the power plant organelle of eukaryotic cells. Let's also talk about the concept of electronegativity, which you may have encountered in chemistry class. So, in OP, electrons are passed down a series of proteins embedded in the inner membrane of the mitochondrion. And each protein in the series is more electronegative than the one before it. So, in simple terms, the more electronegative a molecule is, the more easily it can hold on to electrons. It takes less energy for it to hold on to electrons. So, during the OP process, the electrons move downhill energy-wise, and they release energy as they go. And it's important to remember this in a little bit. Let's look at the components of OP. We have these large proteins, protein complexes labeled 1, 2, 3, and 4. We have a couple of smaller proteins. This one's labeled Q, and this one labeled site C, which are also used in the process. We see where NADH and FADH2 feed their electrons into the chain, and we see the path that the electrons take through the whole, from one protein to the next. In the mitochondrial matrix, the inside here, and the inner membrane space outside here. You also see hydrogen ions being pumped out by these proteins from the matrix to the inner membrane space. We'll get to that later as well. So first steps here, you have NADH and FADH2 feeding their electrons, NADH into complex 1, FADH2 into complex 2. FADH2 is a bit more electronegative than NADH, so it has to feed its electrons in a little further downstream. And as NADH and FADH2 feed their electrons in, they break apart into their oxidized forms, NAD plus and FAD, and also a bunch of hydrogen ions. And they can be used again in the citric acid cycle and glycolysis. The next steps here, the electrons move from both complex 1 and complex 2 to this little Q protein. This full name of this is ubiquinone. It's a small protein that moves about freely inside the mitochondrial membrane. And from ubiquinone, electrons pass to protein complex 3. Then final steps, the electrons pass into this other small mobile protein on the outside of this mitochondrial membrane called cytochrome C or site C. And then the electrons move into protein complex 4. And then the final step, the electrons move into the mitochondrial matrix again, where they fuse with oxygen. And oxygen is an extremely electronegative molecule, so it's a logical last step for these electrons to end up at. And this is why we need oxygen. The oxygen acts as the final electron acceptor of the entire OP process. When electrons fuse with oxygen, the oxygen molecule splits apart into two individual oxygen atoms. The oxygen atoms combine with hydrogen ions to form H2O, water. Okay, so now on to these hydrogen ions being pumped out of protein complex 1 and 3 and 4. And there's usually a lot more hydrogen ions out here in the inner membrane space than there is in the mitochondrial matrix. So this is going to be an active transport process, remembering what you do about concentration gradients. Where does this energy come from? Well, it comes from these electrons that release a little bit of energy as they pass from protein to protein. And that's enough to pump these hydrogen ions out here. Eventually the hydrogen ions come back inside the matrix, but they can't penetrate the cellular membrane. This is a nonpolar layer, and of course hydrogen ions are polar, so they diffuse passively, but they have to use a transport protein, facilitated diffusion. This transport protein is called ATP synthase. And not only is it a transport protein, but it's also an enzyme. In this case, an enzyme catalyzes chemical reactions. In this case, it takes ADP and its enzyme diphosphate and a phosphate molecule and puts them together to form ATP. And that's how the ATP is created during OP. And on average, you get to about 30 ATP molecules. They're created during this OP process, and that's a tremendous jump over the ATP that's created during glycolysis or even a citric acid cycle. Usually you only get about 2 ATPs created per glucose molecule in glycolysis. So here are the references to my presentation. And I thank you for listening. Have a good day.